It is often stated by neo-Darwinists, advocates of neo-Darwinian evolution, that new genetic information arises from a well-known biological process called adaptation. Such famous examples are insecticide resistance, protein production in mutated food, as well as nylon-eating bacteria and the new metabolism in E. coli. Are these examples really the result of new genetic information? Upon further research in genetics, the answer is clearly no. In order to understand why adaptation is not the result of new genetic information, it is important to understand how protein production works. In order for a protein to be synthesized, it must undergo two molecular processes, transcription and translation. To start off with the basics, a protein is a special molecular-like machine that gives out specific and important functionality necessary for an organism. Proteins are made out of complex chains of amino acids called polypeptide bonds. The function of a protein depends on how the amino acids are arranged within the polypeptide as well as the third-dimensional structure of the protein itself. A protein is actually manufactured by the cell, typically the ribosome. Now, in order for the cell to manufacture proteins, it will have to undergo two molecular processes that I mentioned before, transcription and translation. First, the cell requires a series of nucleotide-based sequences, typically written as A, T, C, and G. Although the DNA sequence may look like it encodes something, it is important to know that DNA sequence itself does not actually code anything yet. In order for it to code, it will first need to undergo transcription. This is done by a special molecular machine called RNA polymers, which begins its transcription in a special signal starting region called a promoter. A promoter is a special site where the RNA polymer sits and starts zipping the DNA region, transcribing one of the two DNA strands into messenger RNA. You can think of messenger RNA as little special packets of genetic instructions that code for 1 out of 20 different amino acids, resulting soon into a protein. But in order for messenger RNA to be put into practice, it will need a translating device known as a ribosome that can read those special genetic codes. The ribosome interprets the mRNA strand and starts coding certain amino acids. Not only does it need a ribosomal translation device, but it also needs a special molecular machines that carry certain amino acids to the ribosomal site. These molecular amino acid carrying workers are called transfer RNAs or simply tRNAs. Once the mRNA is interpreted on the ribosome, it codes for 1 out of 20 possible amino acids carried out by the tRNAs and builds them up into complex chains of aminos known as polypeptide bonds. Once the translating process that changes amino acids is finished, a protein is finally formed. Usually, a simple protein contains over 100 amino acids chained together in a polypeptide. And yet, in order for a functional protein to exist, it will need really specific genetic information in the first place. The more specificity there is, the more genetic information exists in the genome and vice versa. Having the wrong code can mess up the protein and either degrade it or render it useless. In addition, the production of a protein can also be regulated by a special molecular protein called a repressor. A repressor sits on the promoter site of the cell and causes the RNA polymers to be limited in terms of transcription, which results into less production of proteins. The stronger the repressor bond is with the promoter, the more stricter it becomes in manufacturing proteins. Now, contrary to the repressor, there's also a special molecular protein called an activator, which enables protein production. Now, let's move on to the arguments that neo-Darwinists often claim that is an increase of new genetic information. One of the famous examples that neo-evolutionists use in defense that mutations can increase new genetic information is antibiotic resistance. They will argue that since bacteria can adapt to antibiotics, it is increase of new genetic information because that antibiotic wasn't present there before. However, little do they know is that antibiotic resistance isn't really new genetic information, but rather a loss of genetic information, where this loss of genetic information has actually degraded the protein. As I have mentioned in my Evolution Fairytale number 1 video, bacteria can actually become resistant to the antibiotic not by increasing new genetic information but rather by losing it. When a point mutation occurs in the bacterium, it changes the receptor site of the ribosome. Since the point mutation has altered the receptor site of the ribosome, it is virtually impossible for the drug molecules from the antibiotics to attach to it and cause the ribosome to behave differently, causing degradation to the bacteria due to the wrong production of proteins. Since the drug molecules cannot attach to the ribosome, it cannot interfere with the ribosome production of proteins, therefore causing the bacteria to be resistant to the antibiotic. Now, even though the bacteria has actually become resistant to the antibiotic, it has actually become degraded rather than fit due to the loss of specified genetic information caused by the beneficial mutation, where this loss of genetic information has actually degraded the protein. In the peer-reviewed PubMed article, Effects of Mutations to Streptomycin Resistance on the Rate of Translation of Mutant Genetic Information, Garner T.K. from University of California, Santa Barbara, and Rice studied the effects of beneficial mutations in streptomycin resistance on the rate of translation of mutant genetic information. To their surprise, they found that some time ago that the mutations that make bacteria resistant to streptomycin actually degraded the ribosomes, resulting the ribosome to be slower than normal in translating some of the RNA codons or messenger RNA into proteins. Due to the slow production of proteins, the bacterium has actually been degraded rather than fit. In addition, in the article Antibiotic Resistance and Its Costs, published by Nat Microbiology in 2010, the author reports that most antibiotic resistance result into a degradation of fitness, therefore resulting into the loss of new genetic information. The author reports that most bacteria, when it comes to environmental stress that become adapted due to mutations, actually lose their specificity and fitness due to the loss of new genetic information. This is because the loss of new genetic information has degraded the production of the protein and caused the functionality of that protein to be either degraded or terminated. So, to sum everything up, antibiotic resistance is not really an increase of new genetic information. It is rather a decrease of genetic information that causes degrading side effects. Now, moving on to the macro scale, let's move on to insecticide resistance. Neo-evolutionists often use insecticide resistance as evidence that new genetic information has increased in the genome of the insects. However, upon further research, it is also shown that insecticide resistance also possesses similar characteristics similar to that of antibiotic resistance. In other words, resulting into degrading side effects due to the loss of genetic information. It is true that scientists have found how a mutation can make an insect resistant to an insecticide. A molecule of the insecticide DDT, for example, acts by binding itself into a specific matching site on the membrane of the insect's nerve cells. In this position, it prevents the nerve from functioning properly. When enough of the insect's nerve cells have DDT molecules bound to them, the nervous system breaks down and the insect dies, which was published by B-Man in 1982. Now, how does an insect become resistant? It's pretty simple actually. It becomes resistant by losing its sensitivity to the DDT molecule. This loss is the result of a mutation that changes the site on the nerve cell at which the DDT molecule binds, which is basically the receptor site, preventing the DDT molecule from binding. In addition, any mutation that spoils the match between the DDT molecule and the nerve cell will make the insect resistant. As with bacteria, resistance can come by reducing the specificity of the protein of the nerve cell, resulting into the loss of genetic information and degrading side effects. In addition, degrading side effects from insecticide resistance has actually been found to be quite common throughout the biological literature. M.W. Rowland from the experimental station in Hertford, England, has reported that mosquitoes that have become resistant to deodorant are less active and slower to respond to stimuli than are other insects. Their resistance to the insecticide is those bought at the price of a more sluggish nervous system. The information loss on the molecular level then appears as a loss in the performance of the insect. So much for the evolution fairytale claim that insecticide resistance actually leads to new genetic information fitness when it clearly doesn't. Now, another false argument that neo-evolutionists use that claim that beneficial mutations can increase new genetic information is genetically modified foods. One such example is a mutation that increases food protein production or the growth of a vegetable. Are such mutations really evidence of neo-Darwinian evolution and increase of new genetic information? To answer this question, we must understand first how mutations actually act on the organism. As I noted earlier in this video, regulatory proteins known as repressors control the making of a protein in the cell. The regulatory proteins exert either a positive or a negative control. When all of them act in the concert, they produce a tight and finely tuned control. Under normal conditions, the rate at which proteins is made is kept at a level that's best for the organism's well-being. The positive control factor tends to raise protein production, and the negative factor tends to lower it. The tension between them makes the control of sensitive to change and permits it to act rapidly and accurately. Mutations that increase the food protein yield in wheat have been discovered in genes that control the making of the protein. They have been found to increase protein production by degrading a negative control factor. One can reasonably suppose that other traits such as the size of vegetables and the amount of milk that cows give are also affected by mutations in this manner. One such mutation after another can progressively increase the rate at which the cells make food production, but they do it at a cost of upsetting the cell's fine control of protein production. A change in any one of many nucleotides in the regulatory gene can degrade the repressor protein gene. Each gene that further degrades the repressor protein will further increase the protein yield. The changes lose genetic information as they reduce the specificity of the regulatory protein. However, mutations in control genes tend to have harmful side effects. They usually disturb the normal balance in the organism's functioning. In humans, for example, too much growth hormone from the pituitary gland can lead to gigantism and to diabetes, which was established by Landau in 1967. Now, another example of degrading regulatory gene is in grains. In grains, selection for a high protein content has been found to result in less starch per seed and less grain per planting, which was published and established by Brock in 1980. Dairy cattle bred, for example, high milk production turned out to be less fertile than normal cattle, which was also published by Hermes et al. In 1987. Similarly, the sizes of plants and animals are normally influenced by hormones that stimulate growth and by other hormones that repress it. The amount of these hormones in the system is determined by the system controls. You can readily see how a degrading mutation can make a plant or animal become larger. The organism will grow larger if a mutation degrades the hormone repressor. It will also grow larger if a mutation degrades a molecule that represses the making of the hormone that stimulates growth. One mutation after another can progressively degrade a repressor protein. As they do so, they will produce a larger and larger organism. A degrading mutation can also make a plant or animal grow to smaller size. A mutation that degrades an activator of a gene for a hormone that stimulates growth will stunt the growth. So would a mutation that degrades a repressor gene for a hormone that represses growth. So, to sum everything up, a mutation that increases the size of an animal or a plant does not contribute to new genetic information, but rather a loss of genetic information. With this loss of genetic information has degraded the repressor protein, or vice versa. You can think of those mutations as genetic switches. If a mutation switches off a growth hormone, then the organism will shrink and be small, and vice versa. So, genetically modified foods aren't really evidence for neo-Darwinian evolution. They're actually based on the loss of pre-existing genetic information that already exists in the genomes of those living organisms. Just like how deactivating an activator protein in the growth hormone will cause the living organism to shrink into tiny size, so too does the same happen when it comes to the growth. Deactivating the repressor protein will cause the living organism to be gigantic, since there is nothing to stop the protein from producing growth hormones. The same can be argued with the adaptation of polar bears. Polar bears are known to have white fur since they can adapt to the cold environment. Now, why do they have white fur? The reason why they have white fur is because they don't have enough melanin. This is because their DNA does not produce melanin, unlike other bears such as brown bears. The protein production that produces melanin has actually been degraded or terminated. Because the protein has been degraded or terminated, the polar bears tend to have white fur. However, this isn't new genetic information, but rather a lost genetic information, since it lost specificity to produce the production of melanin. Now let's move on to the most famous arguments that neo-Darwinists use in defense that beneficial mutations can increase new genetic information. Such examples are nylon-eating bacteria, E. coli. These examples are an example of adaptation in which bacteria can metabolize new things that didn't exist before. One famous example is soya bacteria. Experimenters and evolutionary biologists have discovered that soya bacteria grown in culture have shown that they can live and grow on new substances that they could not originally use and live before. The bacteria will put under strong selection pressure. The experimenters denied them their normal nutrients, which are either Ribitol or Dehydrogenase, and tried to get them to grow on an unnatural substitute. They tried several unnatural sugars and met with success when they used the sugar xylitol. Xylitol is a sugar very similar to Ribitol and Dehydrogenase but does not occur in nature. Mutants appeared that could live on xylitol and cultures were grown from them. Several experiments of this kind have been reported, such as Lerner in 1964, Wu in 1968, Rigby in 1974, Berlin in 1974 too, and Morlock in 1977, as well as Thompson and KRAWIEC in 1983. The experiments moreover showed that not only one but a series of mutations could occur that will first establish a new enzyme to metabolize xylitol, and then progressively improve it. Isolation and culturing of soya bacteria led to a series of 3 mutations. The experimenters started with the wild-type bacteria, which could not grow at all on only xylitol. A mutant soon appeared in the culture and could grow on xylitol. They cultured this mutant and labeled this strain X1. They found that X1 grew on xylitol at about 1/9 the rate of the wild-type on Ribitol. In culturing the X1 strain of xylitol, they found a second mutant appeared that could grow 2.5 times as fast on xylitol than X1. They cultured this second mutant and labeled the strain X2. In culturing X2 on xylitol, they found a third mutant that grew almost twice as fast as X2. They cultured it and labeled the strain X3. The 3 mutations were studied and all were found to be single nucleotide substitutions. These experiments showed that bacteria can sometimes find other ways of getting what they need when their normal nutrients are denied. Moreover, they did it through a series of point mutations. The first mutation enabled the bacteria of point mutations as well as enzymes. 2 more mutations improved the ability. These experiments look like neo-Darwinian evolution in action. At first glance, it might seem so. Because the mutation made the enzyme more active on xylitol, you might think that it made the enzyme more specific. You might therefore think that in this case, new genetic information has been added. But it turns out that the mutation that led to the X2 strain is just another example of a mutation making the enzyme less specific actually. Brian Hartley and his group at the Imperial College in London made a study of this enzyme. They compared its activity in the X2 mutant with that of the enzyme of the wild type. They measured the activity of the enzyme on Ribitol, xylitol, and L-orbital, another unnatural substrate. They found that compared to the wild type, the mutant enzyme was actually less active on Ribitol, more active on xylitol, and more active on L-orbital. Here's a picture of an enzyme comparison of the reaction rates of the 2 forms of the enzyme for the 3 substrates. The plot was made from the numerical data in the Berlin paper. The enzyme in the X1 strain is denoted in the figure by A. The enzyme in the X2 strain is denoted by B. The vertical scale shows the reaction rate, or the rate of catalysis. Note that the mutation that transformed the X1 strain into X2 broadened the range of substrates that the enzyme could catalyze. The enzyme of the wild type A has a reaction rate curve higher and narrower than that of the mutated type B, which is in green. A very specific enzyme will show a high narrow plot of reaction rate. A less specific one will show a low white plot. In this plot, it shows that the reaction rate of B is actually less specific than that of A, and yet B is actually a mutated enzyme that is capable of metabolizing xylitol. The new enzyme could accept a wide range of molecules as substrate than the old one. Therefore, the mutation made the enzyme less specific, not more, and reduce that information in the genome. Because the enzyme lost specificity, its ability to catalyze biochemical reactions has been greatly degraded. Therefore, the enzyme that is capable of metabolizing xylitol is actually not new genetic information, but rather a decrease of it, as you can clearly see in the graph. By comparison, the B mutated enzyme, which is in green, actually has less catalysis than the one in the non-mutated enzyme, which is basically the red one. Molecular biologists have soon discovered that a change of amino acid in a natural enzyme can actually result into being less specific. In a study published by Bone et al. 1989, for example, they changed one amino acid in an enzyme called alpha-galactic protease and found that the enzyme was then able to act on a wider class of substrates. The function of this enzyme is to cut peptide chains at a unique place within a particular sequence of 4 amino acids. Whenever that 4 sum occurs in the peptide chains, the enzymes cut the chain at a unique point within those 4. Bone and his team made 2 changes in the enzyme. Each change made the enzyme less specific. It cut peptide chains at other places in addition to just the one where the original enzyme cut. The changed enzyme was less specific, so it had less information than the original. It was also less useful, as those authors noted and quoted. One of the fundamental functions of an enzyme is to provide specificity by limiting the range of substrates which are catalytically productive. One might have thought that if an enzyme's activity on some substrate increases, it must be because it became more specific to that substrate. But we see that it is not necessarily so. If an enzyme becomes more specific to a substrate, it should become more active on it and less active on the other substrates. Also, the other way around, if it becomes less specific on it, should become more active on some other substrates. The specificity of an enzyme to a substrate is no less important to the cell than the level of its activity. An enzyme that will accept any molecule as a substrate can be harmful. For an enzyme to be useful in the cell, it must limit its activity to its proper substrate. Some of the specificity of an enzyme lies in the kind of reaction it catalyzes, and some lies in the kind of substrate it will act on. The active side of an enzyme can be considered to consist of 2 parts. One part recognizes and binds to the substrate, and the other catalyzes the reaction on it. So it is evident that the X2 mutation inadvertently degraded the place on the enzyme to which the substrate binds. So to sum everything up, we found that beneficial mutations can occur without even creating new genetic information, but rather by losing it. It has also been shown that enzymes that play a role in evolution also seem to be degraded and less specific when it comes to adaptation. Because of this, the burden of proof lies on neo-evolutionists to show that new genetic information is indeed increased when it comes to beneficial mutations. As I have clearly shown, enzymes do not result into new genetic information since they lost specificity. This includes the enzymes from the E. coli, nylon-eating bacteria, as well as others caused by gene duplication and other genetic processes. Because there was no increase of genetic information, it is inconceivable how macroevolution can occur through microevolutionary changes, since those mutations will constantly degrade the genome and the living organism if it keeps on accumulating beneficial mutations that result in the cost of fitness. Thanks for watching.